Hi all. On seeing this image on the screen, are you reminded of some soulful music that lifted your mood sometimes? Or by seeing that baby, are you reminded of your younger sibling who drew your attention? It is this loud music from the microphone. Has it ever disturbed your last minute preparations? Sometimes when your mom just shouted up without actual words, did it send you a message that you had to stop doing what you were engaged into? Hopefully, you can guess what am I trying to bring in focus. Yes, in this session, we are going to learn about the structure and the function of the human ear. Human ear can detect sound waves in a range of 20 Hz to 20,000 Hz. It's not only hearing, this organ is capable of creating balance and equilibrium in the human body. So let's take a deep into each of the parts in the human ear, starting with the external. Now, this is the exposed part of the external ear called pinna. This pinna on the upper side has a cartilaginous ridge and this is called the helix. Just on the opposite side, there is a more muscular part hanging downwards. This is called the earlobe. In between, we have the muscles in the ear, which are the auricular muscles. These muscles cannot contract and relax in a way which is possible in other animals like the cats and the dogs, who can indeed rotate or they can bend their earlobe in the direction from which the sound is received. This is why the auricular muscles of the human ear, being quite non-functional, are called the vestigial muscles. If we see a little in the center of the ear, this part is called the concha. That is one which helps to collect sound and then allow it to channelize inside. Concha leads the sound vibrations into the external auditory meters. Now, this external auditory meters is about 2.5 centimeter in length. If this total length is divided into 3 equal parts, the first 1/3 is lined by hair cells, those who will prevent the entry of any dust particles along with the sound waves. The next 2/3 part is lined by ceruminous glands. Ceruminous glands who secrete cerumen or an oily secretion which traps any microbe that tends to enter into the ear canal, which is also the auditory canal, name for the external auditory meters, and then it doesn't allow any of these microbes to enter inside and infect the tympanum. Here in this image you can see the 3 distinct namings given. This portion with the pinna and the external auditory meters is known as the outer ear. Then comes the middle ear and finally we have the inner ear. But demarcating between the outer ear and the inner ear is a membranous curtain-like structure which we call the tympanum or the tympanic membrane. This is one that separates the external ear from the middle ear. Now, summing up the outer ear, we find we have the pinna which helps in gathering and localization of sound. Then we have the external auditory meters that which is important for sound selection and transmission of these vibrations to the tympanum. And then finally we have the tympanum or the tympanic membrane, which is a thin stretched membrane, highly elastic, that is what serves as demarcation between the outer and the middle ear. This is also found to vibrate in response to the pressure changes or the sound waves that's perceived by the external auditory meters. And the focus is the middle ear. The middle ear, which is shown more magnified towards the right, it consists of three smallest and driest bones in your body. That is an acronym which will make you easier to learn it, is MIS, M-I-S. M for malleus, I for incus, and S for stapes. These three bones altogether has a name called ossicles or ear ossicles. Now, what is their function? These three bones are attached to one another in a lever action. And as we all know, liver helps to get work easier. So, this vibration which was moving from the tympanum now gets these vibrations going with a higher amplitude when it moves through this lever action via the ossicles, and it gets multiplied to an extent of 10 times as to what it carried through the tympanum. The next we have the surface area of the tympanum, which is transmitting it to the ossicles when compared to the surface area of the oval window, where it is about to transmit it into the inner ear, has a large difference. And that difference in the surface area also helps the amplitude of the sound to get multiplied 2.2 times. The oval window is indeed with a much smaller surface area. That's why when the total vibrations are moving there, it gets, magnified to 2.2 times. So a total magnification of the sound which happens in this part of the middle ear is 10 into 2.2 times, that is a total of 22 times. But is it very congenial for the sound waves to get so highly magnified and yet keep the work going very smoothly? There is a question that comes, and for this there is a beautiful arrangement inside the middle ear. We have a eustachian tube. This eustachian tube is a tube-like structure that connects the middle ear to the throat, passage of the throat. And since the vibrations got magnified so highly in the middle ear, this leads to high pressure being developed in the middle ear compared to the external ear. And the demarcator of the middle and the external ear is a very highly flexible thin membrane. So there is a fear that the high pressure may move the tympanum in such a way that it might burst the tympanum and then lead to total hearing loss. But the accommodation of the eustachian tube allows the extra pressure to be let out through the pharynx, and then it doesn't allow any damage to happen to the tympanum by equalizing the pressure on either side of the tympanic membrane via the eustachian tube. This is a complete image of the internal anatomy of the ear. So you can see certain parts beyond the middle ear where you can see the stapes connecting to the cochlea, and then there is the semicircular canals and a vestibule. We will now see the structure and the function of the inner ear in further details. The inner ear is also called the membranous labyrinth. This inner ear is a fluid-filled part of the ear. Outrightly, the sound waves move in the fluid medium, whereas it was moving in the air medium, both in the outer and the inner ear. Now, the three distinguished parts of the inner ear are cochlea, the organ of hearing, or the organ of Corti, is located here. The cochlea looks somewhat like a 2.5 turn of a spiral, which resembles the shell of a snail. Then we have the semicircular canals, three semicircular canals, one anterior, another posterior, and then a lateral semicircular canal. Then we have a vestibule, a compartment-like structure, which has the semicircular canals on one side and the cochlea on the other side. This compartment-like structure is made up of two sac-like structures. A bigger sac-like structure, which is connecting to the semicircular canals, is the utriculus, and a smaller sac-like structure connecting to the cochlea is the saculus. These are also responsible for creating static balance in the body. So, while semicircular canals can establish dynamic balance, the vestibular region of the utriculus and saculus can establish static balance. Now, let's see how each of these balances are created. In the semicircular canals, where we have the ampulla, these ampulla have specialized hair-like structures with stereocilia covered by a gelatinous structure. And here, when the head is moving in the forward direction, this anterior semicircular canal can detect those movements, because when the head is in a nodding state, the gelatinous covering over the stereocilia slips onto one side, and those are detected by the crista present on the anterior semicircular canal, and then they bring the balance by putting the head in the upright position. When it is the posterior semicircular canal, they are capable of detecting the backward movement of the head towards the shoulders. This is also indeed brought about by the slipping out of the gelatinous covering over the stereocilia, who can detect it and when required bring the head back to the straight position. Comes to the lateral semicircular canals with their ampulla and the crista inside, and with the stereocilia of the gelatinous covering in the similar way when it is tilted from side to side. That is, the head gets tilted from side to side in a lateral direction. Here there is disturbance in the gelatinous covering, and that is detected by the lateral semicircular canal to bring it back to the exact central position. So any kind of dynamic condition in the body with movements during that time when it needs to establish a balance, that will be done by the ampulla carrying the crista in the semicircular canals. This vestibule, which is with these two sac-like structures, do lodge a specialized balancing organ too, and they are called the macula. The macula usually has cup-shaped cell which are with the stereocilia coming out of their head region, and the basal end they have the nerves, quite similar to those of the crista-like structure, and these are meant for bringing static balance. What is that balance? When you are in a sitting position or in a standing position pitched at one place for a long time, or that at maximum making a straight movement or a rectilinear movement, it is that you should not lose your equilibrium. That is indeed maintained by the the macula in the vestibule. That is the macula which is present in the utriculus and the sicculus. I have also tried to give you an acronym for an easy remembrance of these functional activities associated with the structures. So I've taken the few letters of the English alphabet towards the end, that is STUV, where ST stands for static. S is also taken for sicculus, U is taken for utriculus, and that they are located in the vestibule. So the sicculus and the utriculus located in the vestibule can establish static balance in the human body. Now let's go further into the cochlea, because this is very important for understanding how we hear the different notes and the loudness of sound. Cochlea further contains the organ of Corti, which is highly responsible for the process of hearing. When we go through a cross section of the cochlea, we find three chambers are distinctly visible in its inner structure. The part which is connected to the vestibule, it is called the scala vestibuli. The scala vestibuli is filled with the fluid called perilymph. We have another chamber called the scala tympani, which is the lowermost chamber. This is also filled with perilymph, both of them being shown in the blue colors. In the center, we have the scala media. It is filled with end lymph, and end lymph has higher concentration of the potassium ions because it is supplied by the stria vascularis from time to time. This difference in the ionic concentrations help in the passage of electrical waves to carry the impulses of sound for the process of hearing. Now let's see, when the sound is moving from the fenestra ovalis or the oval window, which gets the vibrations transmitted by the stapes, it enters into the perilymph of the scala vestibuli. This causes disturbance in the fluid and it gets carried through to the scala tympani. The scala tympani is separated from the scala media by a stiff basilar membrane. So this basilar membrane is set into vibration, and sitting on the basilar membrane is the organ of Corti. This organ of Corti is made up of sensory cells with tufts of hair-like structures and at the tip having the stereocilia. They are very closely in contact with a very sensitive membrane called tectorial membrane. Now, if we look further into the details of this sensory cells, what we find, the fibers near the upper end, these fibers near the upper end of the cochlea, they resonate to the lower frequencies, whereas the fibers that are present on the basal part, that is close to the oval window, they will resonate to the lower frequencies of sound. And these sensory cells, which are located right on the base of the basilar membrane, they will be resonating in bigger numbers when the loudness of sound is more, and less of these sensory cells will be bending if the loudness of sound is less. Finally, these, when they start vibrating and bending, thus touching onto the tectorial membrane, they release out a neurotransmitter because of the influx of potassium ions, and that indeed transmits an impulse through the nerve endings that emerge out at the base of it, and finally get carried via the auditory nerve and the cochlear nerve to the central nervous system. Now, this is an overview of the different parts of inner ear, showing the transmission of the sound waves and the change of these vibrations into electrical waves for further interpretation through the brain, where sound waves come in the ear medium from the external as well as the middle ear. It moves through the fluid medium in the inner ear, and then in this inner ear through the organ of corti, they are detected with the differences in frequency and loudness, and then transmitted via the auditory nerve to be detected and interpreted through the central nervous system in the brain. The total auditory pathway is shown in the diagrammatic manner here, starting from the outer ear, moving through the middle ear, then finally coming through the inner ear and passing to the brain through the auditory nerve. We humans have taken hearing for granted. All our normal activities require hearing. The ability imparts tremendous survival strategies for animals. In addition, the human social structures rely largely on speech, communication, and activities associated with hearing, which requires the sensitive, rapid processing of the sound energy that the normal inner ear provides. So, happy learning. Thank you for watching. Please do like my video. Share it with your friend if you find it useful. And do not forget to subscribe to my channel. Thank you. Happy learning.